All right. Hi, everyone. Welcome to week 8 of Biochem. I hope your exam went well, and I hope you had a great spring break. So this, or I guess I should say from this point on, we're kind of shifting gears, and we're going to stop talking about, generally, here's how enzymes work. Generally, this is what happens. This is the last chapter of those generalities. And then we start shifting into specific things. So we're going to start talking about glycolysis. We're going to talk about the pentose phosphate pathway. Glycolysis, I just said glycolysis, gluconeogenesis, there you go. The tricarboxylic acid cycle, the external transport chain, etc. But we're kind of done with the usual, if that makes sense, and we're moving into new stuff. So I guess let's jump into the first question. So before spring break, we were talking a lot about enzyme kinetics normally, right? Normally, an enzyme will do this. Normally, if it follows Michaelis-Menten kinetics, it'll do this thing. But even with enzymes that follow Michaelis-Menten kinetics, you can have regulation. You can have things that kind of break Michaelis-Menten kinetics, if that makes sense. And so, sorry, excuse me, we have what's called regulation, right? And so regulation, I'm highlighting, that's fine, let's highlight. So regulation, that's one of the key words here. So this could be either activation, which is exactly like it sounds like. It makes the reaction increase, so plus, or inhibition, which also is exactly as it sounds. And we generally draw minus signs for it shuts down the reaction or it slows it down. Okay, so that's the key thing. A regulator is the thing that does regulation. I don't know if that needs to be said. And an activator or an inhibitor are the thing that activates, the thing that inhibits. Fairly self-explanatory. Okay. Next is, it's happening through the enzyme. So this is something that is also not super complex, but I have seen people mistake it. So the regulator binds the enzyme, it doesn't bind the substrate. Okay. And finally, it's at the site other than the substrate bindings. Okay. So this part's all true. Okay. I should say the part that I drew the arrow on, the site other than the substrate binding site. So great example of this, well, not a real example, but a drawn example of this would be if I have this enzyme and I grab an inhibitor, which I'll be using red for. So inhibitor, and it binds right here. Well, what that could do, and that could change this thing's shape, like that. And so now my substrate, which I'll label S, can no longer fit in this spot, right? Sorry, enzyme. There we go. And so that's kind of the key to a site other than the substrate binding site. So when I think of that, I think of it, so if we think back to inhibitors, the way we learned them before spring break, we learned competitive inhibitors bind to the enzyme and compete with the substrate. This is not like a competitive inhibitor at all. Okay, competitive, there we go. It's not like that at all. Uncompetitive and non-competitive bind to the enzyme substrate complex in some way, and they kind of mess with things there. This also, I think of it fairly similarly to uncompetitive, because an uncompetitive will bind to the enzyme substrate complex. However, that being the case, or I should say, that said, it's really its own unique thing, right? The allosteric inhibition is not like the inhibitors we've been talking about before. Okay, so that was me rambling on for 4 minutes about regulation without actually answering this question. The key word here is covalent. Okay, are we actually binding covalently? And the answer is no. It's non-covalent binding. Non-covalent. Okay. So reminder, covalent binding is like this sulfur bound to this hydrogen, bound to this other hydrogen, whatever. These are the covalent bonds. So a non-covalent bond would be something like something like a hydrogen bond, some kind of intermolecular force is non-covalent. So because of that, this one is false. Okay. All right, the next question, which of these amino acid side chains cannot be phosphorylated? Okay, so we talked about phosphorylation, I believe, before the week before spring break, so week 7 of our reviews. And one of the things that I mentioned with phosphorylation is it's one of those post-translational modifications, right? So it's one of those things, once I've made my protein, I can add a phosphate group to it. That phosphate group ends up being negatively charged. That negative charge affects whether for example, my serine, assuming serine can be phosphorylated, can travel through the cell, right? If it's negative versus neutrally charged, maybe that'll affect something, and so on and so forth. It can also affect binding. It can activate or deactivate. Phosphate does about 1 million things, and so it would take too long to go through them all. You'll be learning more throughout the semester, but that's kind of the big thing. But anyways, so let's draw these structures because that'll really help. So serine, I know, is an OH. I know it's got some other stuff like a carbon. Aspartate is something carboxylic acid. Glutamine is kind of the same with one extra carbon, except it's an amide. And then tyrosine is this ring with an OH. Okay, so there's one thing I want to point out. What do they have in common? And this is something I always suggest. If you're not sure, what do they have in common, right? And so when we go back to week 7, and I have to go to the top. Oh, I apologize. It was week 6. My mistake. So when we go back to the top with week 6, was it week 6? Yes. Nope. Okay, it was a while ago then. Let's go back to week 5. Okay, it was not week 5. Okay, then I know I put the slide up. I don't remember exactly when. I apologize for that. I thought it was last week though. Okay, that's fine. But the key here is, skipped a lot ahead. Look for the similarities, right? In this case, we see 3 OHs that are similar. That makes this NH2 kind of the odd one out. Phosphate groups, the other thing is if you're not sure, draw it out, right? I know a phosphate group looks something like this. It can have, various numbers of hydrogens, but every single attachment is an oxygen, right? And so if I don't get an oxygen, there's a problem. And so because of that, the answer to this one is glutamine, right? Glutamine cannot be phosphorylated. Or I should say, the glutamine side chain cannot be phosphorylated. If you have glutamine on the C-terminus, I believe it can be, and that's a completely different question. Okay, pro-insulin. So we've talked about allosteric regulation very briefly. We've talked about phosphorylation and generally post-translational modification. This is another type of regulation. So pro-insulin is a great example of this. And insulin, or I guess I should say, pro-insulin is what's called a zymogen, or a proenzyme, exactly like it sounds like. And so what happens is, and I'm going to write that down, these zymogens are precursors, okay? So it's pro-insulin, I think of it as just pre-insulin, precursor, excuse me. So it's right before insulin is this thing. So what it ends up looking like, and I'm going to end up using 3 different colors for this, is peptide part A. Actually, hang on, it goes like that. I apologize, let me redo that. Peptide part A, part, no, I want to do right with this one, I apologize. Part B, and peptide part C. Okay. All right, and I'll go ahead and label those. So A, C, and B. Okay. So this is pro-insulin. So now what I'm going to do is I'm going to use this lasso, and I'm going to copy this. I'm going to paste it right over here. Did it paste? I can't tell if it pasted. Give me one moment. Oh, my OneNote is not responding. I apologize for the delay. It might take a moment. Regardless, while it's loading, I'm going to try to speak. So pro-insulin has what's called a connecting peptide. Okay? So what this ends up meaning for us is a connecting peptide is exactly what it sounds like. It connects 2 parts. Okay, so part B is the connecting peptide because it connects A and B. So let's think about this in terms of what's going to happen. So this pro-insulin gets translated. So now we have this big long peptide chain that's A plus B plus C. Okay. Then when it's, right after it's been translated, it's going to start folding into whatever the most stable configuration it can take is, right? So just like we learned about alpha helices and beta sheets and that kind of secondary structure stuff, this is going to try to do the same thing. Does it make an alpha helix? Does it make a beta sheet? Whatever. Oh, here we go. Okay, I apologize. I think it's finally resolved itself. So let me try to do this again. So now we copy this and we paste it. Okay, awesome. And I'm going to move this copy down here. Okay, problem solved. I hope we're good. Okay. So anyways, so yes. So it's been translated and it takes on this shape. And the reason it takes on this shape is because I need green, okay, is because there are disulfide bonds here, okay? So this we're gonna label pro-insulin is this one. And so now once these sulfide bonds have formed, so basically B is there to connect A and C so that they can form that sulfide bridge together. We don't really need B anymore. So we're just gonna get rid of that. Oops, I didn't mean to delete C. We're gonna get rid of that part. And I didn't draw the sulfide bonds here, but there are some sulfide bridges, and so this becomes insulin. Okay. In other words, pro-insulin is insulin, except it's got a connecting protein that we eventually get rid of. Okay, so D is the exact opposite. Insulin is pro-insulin, except without a connecting peptide. And once again, B is also exactly wrong. It's the inactive precursor form of insulin. And A is also incorrect because it's insulin with peptide bonds and also an extra subunit. Okay, all right, and I'm going to zoom out a little bit so we can move to the next question. Excuse me. All right, so this is a question where we're going to start getting into some of the actual notation, some of the words that we need to learn and that we need to understand. Okay, so when I get a question like this, I get overwhelmed. I do. There's just too much going on. There's a bunch of letters, a bunch of words. It's too much. So I try to draw a diagram. So A reacts to form B. Okay, cool. A reacts to form B. I am done with that part. Okay, and a reaction catalyzed by enzyme E1. So I'm going to put E1 here. Okay, cool. Done with the entire first sentence. So up till here we're good. Okay. B then forms C in a reaction catalyzed by E2. Okay, cool. Now we've just kind of translated this. Oh, and I didn't write C. There we go. And now that's the final product in this pathway. So this pathway is done. But C can bind to E1. Okay, so I guess I'm going to draw it in a different color. And I'm going to draw it in blue. So generally speaking, I'm going to draw green as activating, red as inhibiting, blue as we need to figure that out. Okay, all right. Okay, so it binds to E1. We're good up till here. It speeds up the rate of B formation. So blue, we're gonna go green. Or I kind of spoiled it. Blue speeds it up, so I'm gonna change it to green. Okay, and I'm not gonna actually erase and redraw, but I'm just gonna say this is now green. Okay, so let's think about this. So allosteric is a really cool term to keep in mind, right? And again, it binds to an enzyme at a site that's not the active site. Great. Excuse me. Okay, so let's go through some of these terms. So first of all, we know it's not an inhibitor, so we're going to get rid of, and I should switch back from green. Let me do that real quick. Okay, so I'm going to get rid of anything that has inhibitors in it, and we're going to talk about inhibitors in a little bit, and I'm going to go through each of these in time. C is not a regulatory enzyme, right? C isn't an enzyme at all. The enzymes are up here. So just like that, we can get rid of bullet point, of answer choice C, because product C isn't, I should say. I apologize for the confusion there. Okay, and so that leaves D. And D is correct. But I want to talk about each of these terms slowly to make sure that we understand each of them. Okay, so an allosteric inhibitor and feedback inhibition, or I guess I should say allosteric regulation, is kind of the top of this tree, and then it branches out into allosteric activation and allosteric inhibition. Okay, and I apologize, I'm inhibition, that's fine. Okay, from allosteric inhibition, we branch out again to just regular old inhibition and then to feedback inhibition. So feedback inhibition is basically what happened here, except if our blue arrow was red, right? In other words, let me clarify that. So allosteric or feedback inhibition is where A goes to form B, which goes to form C. Cool. Then when we have enough of C formed, C says, okay, stop that. We don't need any more. Shut this down. Okay? And that stops A from being used up to make C. And we're going to see this. It's a very common thing, feedback inhibition. We're going to see it in glycolysis when we get to that glycolysis unit. We're going to see it in most of the units coming up. Okay? That's feedback inhibition, which is a type of allosteric inhibition. Okay? Now, I said the regulatory enzyme, well, enzymes are the things I highlighted in green, right? The regulatory enzyme is specifically this one. And the reason is the regulatory enzyme is the enzyme that, I guess, let me rewrite that. It's an enzyme which represents a point when our metabolic pathway is regulated. In other words, it's an enzyme that's regulated. It's exactly what it sounds like, right? Enzyme regulated. I mean, it's in the name, okay? Finally, the allosteric activator goes through allosteric regulation, just the same as the rest, but it's an activator. In this case, because we ended up with the green arrows, we're dealing with an activator. If it were an inhibitor, this would be both A and B. I would pick B as the most correct answer, okay? All right. So that's, excuse me. So that's kind of the main ways we talk about regulation. There are quite a few more, obviously, but those are the bigger ones that I really want to focus on. Now let's talk about kinetics, or rather, let's talk about kind of how regulation affects kinetics, okay? So right off the bat, this is the standard Michaelis-Menten enzyme kinetic curve, right? It's got that beautiful kind of first order at first, and then it goes to second order. They label Vmax, 1/2 Vmax, Km, all the rest. Great, okay? And the curve, like we mentioned, gosh, in like week 6 or something, is a hyperbolic curve, right? That's what that kind of sideways U shape is. It's hyperbolic, okay? However, regulatory enzymes do not show this curve, okay? So this is for something that obeys Michaelis-Menten enzyme kinetics. Now we're going to talk about regulatory enzyme kinetics, okay? So they're different. Enzyme kinetics, and I apologize, I'm a bit of a slower writer. So regulatory enzyme kinetics show what's called a sigmoidal curve, okay? And so I apologize, I'm probably gonna have to undo a few times because I'm not the best artist. It kind of looks like that. I think of it as an S shape, but I'm not very good at drawing the S shape, so it's something like that. And the reason it shows this S shape is we can think of it as the binding of the first molecule will speed up binding of subsequent molecules. So let's just look at, and I'll do it in blue, this first section, okay? This reaction's going great, very fast, very, very effective. This is very slow, okay? Why? Because it's just, the first binding is the most difficult binding, okay? All right, as soon as that first one binds, we show positive, like the reaction rate shoots up, okay? So it took, if we think of it, from here all the way to here to get to this height. It took the cooperative enzyme from here to get to that height. And so the change in rate is a lot faster as soon as the first one binds, okay? And that's what cooperativity means, okay? So cooperativity equals binding of substrate 1, which I'm just going to say is the first substrate, facilitates subsequent substrate binding. Okay. So in other words, if this enzyme can have, let's say, it's got 3 active sites, okay? So the first active site binds to substrate 1, or binds to the first of these substrates, and they're all substrate A, right? So they're not different. As soon as active site 1 is filled, active sites 2 and 3 become more open, and so it very quickly speeds up the reaction rate and catches up, okay? Right. Now, I don't know about you guys, but that doesn't actually explain much to me. Like in my mind, I'm like, okay, what are you saying? And so scientists agree, don't worry, and they've come up with 2 models to try to explain this cooperativity, this regulatory enzyme kinetics. And those models are the MWC model and the KNF model, okay? And in both cases, it stands for some people's names who we don't super care about right now. And so I'm going to call them MWC and KNF. I may occasionally call them by a different name, in which case I'll explain that name to you guys, in depth and more slowly. But let's go through them, okay? So in the MWC model of allosteric regulation, which of the forms of the enzyme is able to bind to a substrate? Okay, and the correct answer to this relies on, well, what are these forms of the enzyme? Okay, I'm going to switch back to black. So we can think of the enzyme as having a, as you guys can tell, a tense and a relaxed state. Okay, so for the sake of me not being the best drawer or artist, whatever, this is going to be our enzyme. Okay, and this is in what's called R0, its relaxed state. Okay, and this R0 is in equilibrium with what's called the tense state. Okay, so the tense state is going to have little, I don't know what you should call them, little circles in the middle of their active site. So this thing is what differentiates the relaxed and the tense states. Okay, and they're in equilibrium, and the equilibrium constant here is called L. Okay, so again, for the sake of convenience, because I am a terrible artist, this is our substrate. Okay, even with my pretty poor drawing, I don't think it needs to be explained in too much depth that the substrate will fit in the relaxed state. So the answer to this one is B. But there's a lot more to the MWC model. And the reason is because the MWC model basically says, or I guess I should say, let's start off by defining L. Okay, so L is obviously the equilibrium constant, but there's more to L. And the reason there's more to L is because it obviously is product over reactant. So if we think of T0 as the product and R0 as the reactant, L is almost always large. Okay, and I apologize, I'm going to be scrolling down here. L is large. Okay, in other words, T0 is almost always greater than R0. Okay, so again, let's think about what that means. The tense state, or I should say, enzymes tend to exist in their tense state according to the MWC model. Okay, this is not according to like reality. No one's done like, or I'm sure people have done research on it, but I mean like, this is not something that we're saying as gospel fact. We're saying the model is suggesting that the tense state is the state that they're most often in, but that the substrate has a higher affinity for the relaxed state of which there's less. Okay, in other words, the substrate binds with a higher affinity to the less abundant form of the enzyme. Okay, all right. So now let's talk about why that or I guess like how we can justify that. Okay, so over here to the right, and we're going to need quite a lot of space, so I'm going to basically redraw my model. Okay, and I'm also going to simplify it a little bit. Okay, so that is once again R0. It's in equilibrium, and I apologize, I don't mean to draw crooked, I just kind of do. And it's in equilibrium with T0, okay? And once again, here's my substrate. Okay, that was weird. Here is my substrate, all right? Well, let's think about this. As soon as this substrate binds to R0, we get a new equilibrium, okay? I mean, if we think of it in the same way that we thought about, you know, E plus S goes to ES, when I added an inhibitor, I drew equilibrium arrows going this way with my inhibitor to get my EI complex. This is the exact same thing, except now it's with a substrate, okay? Right? And the equilibrium will be affected in the exact same way, right? So now what have we done? When we introduce substrate here, oh, and I apologize, I should mention, this is called R1, so relaxed state 1 rather than R0, okay? So again, let's go through it kind of step by step. If we just bound some of R0 and turn it into R1, we've effectively decreased the concentration of R0. I could put that in there. There we go. Well, when we decrease the concentration of R0, looking at this equilibrium, what's going to happen based on Le Chatelier's principle? Our reactions, or this equilibrium will shift to the left. In other words, by binding R0, we get more R0 from T0, okay? So that's how so we talked about cooperativity kind of in the abstract. Binding of substrate, the first substrate facilitates subsequent substrate binding. This is one of the models that kind of tries to explain how that happens. So when we bind the first substrate, we lose our free enzyme, the relaxed state, and so more of it is formed from the tense state to make up for that based on this equilibrium. Okay? All right. So that's the MWC model in a nutshell, and we're going to be talking more about it, don't worry. But for now, let's shift to the KNF model. Okay? Oh, and I kind of already answered. Oh, no, I didn't answer number 8. That's fine. Okay. So the KNF model is called the sequential model. And actually, I apologize. I need to erase where I talked about L because I need a bit more space. Okay. So the KNF model is called the sequential model. Okay. And the reason it's called the sequential model is because it happens in sequence, right? I don't know if I hope that was kind of self-explanatory based on the name. But what happens in sequence? Okay, so I like to think of it as something with 4 subunits. Okay, or actually 3 subunits for this example. Okay, and we're going to keep it color-coded. We'll use black. Okay, so we have 3 subunits. Okay, the first step. Let me redraw that. The first step is we're going to add a substrate. Okay. When we do that, we get this, okay? So the substrate causes conformational change in the subunit it bound to, okay? Then that causes a conformational change in the next subunit, okay? And I'm gonna make that a blue circle, and the circle is empty. So this, I'm gonna kind of shade around it, and in the next one, I'm gonna shade it completely to say that it's bound to something now. Okay, so this is blue. Okay, what does the blue enzyme mean? And I guess I could label this subunit 1 or M1 or M2 or something, but I'm not going to. But effectively what's happening, and as you guys can tell, we're going to talk a lot more about it in a little bit. So I'm not going to answer this question completely. I'm not going to answer a ton about the KNF model yet because we're going to get there. But it's called the sequential model because substrate binds affects, we'll call it M3, right? Just going M1, M2, M3. M3 then affects M2. M2 then affects M1. It's happening in sequence, okay? Whereas the MWC model, I don't think I asked this question, no I didn't. The MWC model is called the concerted model, right? So it's happening in concert, it's happening all at once. Concerted model. In other words, there's not like half relaxed, a quarter relaxed, subunit 2 relaxed, whatever. There's relaxed state, tense state. There's not a middle ground there, right? Here there is middle ground, right? Because there's maybe this one is relaxed and this one's tense or vice versa, whatever the case may be. And so the KNF model is very clear. It happens subunit by subunit. The MWC model, it happens all at once, okay? So that's the difference between the sequential and the concerted model, okay? So these next 3 questions I'm going to answer with diagrams, and I'm going to zoom out so we can see the diagrams. So just a quick, read the questions. Question 8, how does an activator increase substrate binding in the MWC model? And question 9, how does an inhibitor decrease substrate binding in the MWC model? Okay, and I'm going to answer both of them with this figure. Okay, so this figure is exactly what I drew at the beginning, or I should say this part of the figure. I don't like highlighting. I'm going to make it, okay. So this part of the figure is exactly what I drew, except admittedly a lot more poorly, which is fine. Okay, so now let's talk about what's actually happening here. Okay, this equilibrium is that L that we defined above, right? Okay, when I add an activator, the activator binds to the relaxed state of our enzyme. Excuse me for a moment. Okay, sorry about that. Okay, so the relaxed state gets bound to this activator. In other words, we lose some of R0 to create R1A. Okay, so R1 is bound to this state, and activator it's bound to an activator. Substrate would be if it was bound to a substrate. So this is R1A. So now we've made some R1A. And in doing so, and actually I just realized I should keep everything the same color. So I should separate colors out. So when we talk about activators, I'm going to keep it in green. So when that happens, Well, now R0 decreases. When that happens, Le Chatelier's principle tells us this equilibrium shifts to the left, and we get more R0. And now both R0 and R1A can bind to substrate to give us this R1AS and give us some R1S. Okay. In other words, an activator does the same thing as a substrate with positive cooperativity by binding to the relaxed state to decrease the concentration of R0 relaxed state, which then shifts the equilibrium to cause more of R0 to be formed. So then the sum of all of these relaxed states increases. So this diamond goes up. Okay. All right. Now that's number 8. Now let's talk about number 9. How does an inhibitor affect things? I'll switch to red so that we can talk about the inhibitor. So everything we've been dealing with has been binding the left side of this equilibrium, right? Now we're going to finally deal with the tense state. And the reason we deal with the tense state is because an inhibitor wants to make more of the tense state. So when an inhibitor binds to the tense state, it creates T1I, so T1 inhibitor. When that happens, we do the same thing to the tense state as we did to the relaxed. We've decreased its concentration. Therefore, Le Chatelier's principle is going to decrease the concentration of relaxed state to make more tense state to reach equilibrium. Okay? So that's kind of a lot. So let's take it slower, and I'm going to erase this diamond because I think it's just kind of in the way of everything, right? So effectively, I'm going to kind of simplify things. An activator increases sum of R0 plus R1 plus R whatever. So in this case, it's all R1, but just in case there are different notations, okay? By decreasing or by transferring R0 to R1, then making more R0. Okay? An inhibitor does the exact opposite. So an inhibitor makes more T0 plus T1 plus however many T's there are, 10 states, by transferring T0 to T1, which then transfers R0 to T0 and decreases R0 concentration. Okay, that's kind of the big part of it. Okay, so now this is great for positive cooperativity, right? So I told you cooperativity is binding the first substrate facilitates subsequent substrate binding. That's about 90% correct, and that's how we mostly deal with cooperativity. But there's positive cooperativity and there's negative cooperativity, just like there's positive and negative with everything, right? So positive cooperativity means that the first substrate increases subsequent substrate binding, and negative cooperativity means that substrate 1 decreases subsequent substrate binding, okay? So that's kind of hard to draw with the MWC model. Let's see how KNF addresses it, okay? So let's start with just what we're looking at here, okay? So we have 2 subunits, right? And I'm going to call them M1 and M2, again, for the sake of convenience, M1 and M2, okay? The substrate binds to M2. So this is M1, just like normal. This is M2 plus substrate. That causes a sequential, right? It happens sequentially. It transmits this conformational change to convert M1 from a square to a diamond. So I'm going to call it M1 prime, okay? So now let's talk about cooperativity. So this figure confused me so much when I was taking biochem. I didn't get it, okay? What I'm going to tell you guys to do is ignore the right side. Look at just the first part, okay? So this is comparing relative affinity. And what that means is it's basically saying, does the substrate like this one or that one more, okay? So this is, once again, M1 in both of these, M1. So what this is saying is that M1 prime has a higher affinity for substrate than M1. That's what this diagram is saying. In other words, the conformational change activated M1. That is positive cooperativity. Okay. This is the only type of cooperativity that we can show with the MWC model. However, the Knf model, oh, and I guess I should have done that in green, so I'm going to do that instead. Okay. And I'll switch to red for this next one, even though for consistency, I really want to just keep it in black and just do a red star, but that's okay. We're going to switch to red. The next one is saying, oops, there we go. This is M1 prime, and this is M1. In other words, the conformational change decreased the affinity. Decreased affinity. So affinity went down. This is an inhibitor. This made, or I should say, this is negative cooperativity. Negative cooperativity. Okay. So the Knf model is more easily able to show both positive and negative cooperativity, whereas the MWC model struggles to show negative. Okay, that's kind of why we have both, is because they both are valid, by the way. I want to start off by saying, because I don't know if I explained that. Yes, there are models that we believe describe things, and yes, they're different models, but the way that I think it should be phrased is each model has its uses for specific enzymes in specific situations, okay? And we're going to talk about a protein. I don't know if it's next or if it's in like 2 or 3 questions. I'm afraid I don't remember. But we're going to talk about a protein that shows both MWC and KNF, characteristics, okay? Okay, yeah, not yet though. Getting there though, in a few minutes. All right, so which protein shows sigmoidal oxygen binding kinetics? So we have myoglobin and hemoglobin, and hemoglobin is what we're going to be talking about for most of the rest of this review. Before we get into hemoglobin, I want to start off by saying, hemoglobin is a tetramer. Okay, so let's kind of define each of these. Hemoglobin, tetramer of alpha 2, beta 2. Okay, so it's got 4 subunits, 2 alpha subunits, 4 beta subunits. Okay, myoglobin is a monomer. Okay, does it matter? Not yet, but it will. Okay, and then more specifically, hemoglobin carries oxygen in the blood, myoglobin carries oxygen in the muscle. Okay, so the reason this is important is because structure dictates function. So if they're going to have functions that are different, they should have different structures. And let's see how their binding, like the kinetics of this binding is affected by their, I guess I should say affects how their binding kind of indicates is what I'm trying to say, why they're used for what they're used for. Okay, so let's start with myoglobin. So that's the blue curve at the top. Okay, it's very Michaelis-Menten-esque, right? It increases, and it levels off. Cool. So let's think about this, right? Because it's found in the muscles. Oh, I guess I didn't write that. Muscles, blood. Okay, so in the muscles, it's going to get saturated with oxygen, even with like a very, very low partial pressure, right? That's awesome. So binds O2 this is for myoglobin, very rapidly in almost all cases. And I guess instead of rapidly, I probably should have said tightly. Okay, hemoglobin, on the other hand, it shows that kind of sigmoidal thing. So this is that S shape that I was trying to draw. It starts off relatively flat, and then it shoots up, and then it flattens again. Okay, that's the shape that I was trying to draw that I think I kind of did poorly. That's okay. So let's think about this. So they label working muscle, resting muscle, venous pressure, and arterial pressure. So let's think about the arteries. The arteries carry oxygen to the muscles. Excuse me. So no wonder they have a high partial pressure of oxygen, right? They've got a ton of it. Okay. Then we travel to the muscle. So we're going to travel from the artery to, we'll say, resting muscle / working muscle. So I'll go right in the middle. Well, resting muscle, I'm not going to transfer a ton of oxygen to. And that makes sense, right? Because if it's a resting muscle, it doesn't need oxygen. It's not using the oxygen up. If we go to working muscle, it needs that oxygen. Or I should say, hemoglobin has a very low affinity for oxygen, so it's going to let it go, right? So percent oxygen saturation in resting muscle is like 90%. In working muscle, it's 10%. And that makes sense. The resting muscle doesn't need it. The working muscle needs the oxygen a lot, and so it's going to pick it up at a much higher rate, and so hemoglobin releases it at a much higher rate. Okay, and then in the veins, so now that it's traveled to the muscles, it's going to travel to the veins to get more oxygen. Some's bound, some's not a huge deal. Okay, but then it travels back to the arteries and it picks it up, right? And so that's kind of how it works. Okay, so hemoglobin is the protein that really does show both MWC and KNF binding pattern, I guess is the word I'm going to use. Okay, and here's why. Okay, I'm going to do my best to go slowly because this is not the most easy concept in the world. So I'm going to start off by just kind of defining what we're dealing with. Okay, so I'm going to use gray and blue because they're two neutral colors. Okay, and I'm going to use squares and circles just like we've been using above. Okay, so blue is the alpha subunits, gray is the beta subunits. Okay, all right. So the first step is exactly one. So yes, it can hold 4. We're going to say 1O2 binds. And the first thing it's going to bind to is the alpha subunit. So it's going to bind to the alpha subunit. The other alpha subunit stays the same, no big deal. Beta subunits are themselves, right? Nothing's changed with the betas. Okay, great. I mean, that's awesome, right? However, as soon as this one binds, that induces, that transmits a conformational change to the second beta subunit. And so the first beta subunit looks like this. The second beta subunit looks like this. And I'm sorry, I've been saying beta, I meant alpha, I apologize. The alpha subunits, so let me restart that step because I might have misspoken for the whole thing. So the binding of oxygen to the first alpha subunit induces or transmits a conformational change into the second alpha subunit, which is why I drew it as a circle in the third drawing of this thing. The beta subunits completely unchanged. Okay, awesome, makes sense, we're good. Okay, then second oxygen binds. Okay. And once again, it's exactly 1O2, and that's going to bind to the second alpha subunit. Okay, all right. And we're going to talk about why all of this is happening kind of in 3D, but first I want to draw the model. Okay, all right. And then we get this and this. Okay, all right. So now I want us to think about this. I don't know what just happened. Let me draw that. There we go. Let's think of this as the T0 state in our MWC equilibrium. Okay, and on the other side of the T0 state is the R0 state. So let's go ahead and draw the R0 state. So I have, that's a very poor drawing. My two bound alpha subunits, which are still not looking very good, and my two beta subunits now as circles. Okay, so why are they circles? Because they've now increased, so both of them had that change. Okay, all right. So, excuse me. So, right. Now, more oxygen can bind, and I'm going to go ahead and just draw 2 oxygens entering this system because it doesn't really change anything. The reason it doesn't really change anything is we have this circle, and then the 2 gray are now also circled, right? We care about the step-by-step after this point. Okay, so let's dissect what's happening here in depth in terms of what's actually happening, what the models are suggesting, etc. Okay, so first, okay, it's important to note that the first binding of, the first binding of an oxygen, or before the binding, sorry, I apologize. Let's talk before the binding of oxygen to hemoglobin. So here, let's change that to black so we can avoid sticking to one color over another. So here, the iron in hemoglobin is kind of sticking out a little bit. And what I mean by the iron in hemoglobin is, so the structure of hemoglobin, I'm trying to figure out the best way to do this. I'm going to take this area over here. So the structure of hemoglobin is basically an iron ion in the center with, I believe, a +2 charge, and then 4 nitrogens. Was it 5 nitrogens? 4 to 5 nitrogens. I'm afraid it's 4 nitrogens. Kind of surrounding it, giving it electron density, and then they're all in like 5-member rings, and it's just kind of really complicated. We don't care about it from here. Right? The big thing to remember is it's 4 nitrogens, 1 iron. Okay, how does that look? So if we were to look at it from flat on the page, the 4 nitrogens are flat. So I guess I'll draw it kind of like that. The iron is sticking up a little bit, so they all have to go up. Okay, so what does that have to do with anything? And it's a good question. But what happens is the first binding. So let's come back over here. So iron above plane, nope, wrong. Is it plane of heme? Okay. As soon as the first one binds, iron is now in the plane. I hope I'm using the right plane. I apologize if I'm not. Right. So this affects the second hemoglobin. This affects the second alpha subunit of hemoglobin. This is textbook KNF, right? This is exactly what we drew up for the KNF model. Okay, right? Now let's talk about the rest of this. So why is it that when we get to here, it switches to MWC, right? Because this is also textbook MWC. That equilibrium right there, I've highlighted that on about 6 or 7 different MWC model drawings. Why does it switch? And the reason that it switches is, again, what's happening in 3 dimensions, right? And the answer to that is, as soon as the second oxygen binds to the second alpha subunit, there ends up being a fairly large conformational change. And basically what it does is it opens the beta subunits a little bit, the active sites a little bit more, and makes both of them more available to oxygen. Okay, so what happens here is a conformational change. It rotates about 15 degrees, by the way. I don't know if I said that. It rotates 15 degrees, which is fairly large, even this is a giant protein. Opens active sites of beta subunits. Okay. And when that happens, oxygen can bind to either of them. Both of them end up having a high affinity for oxygen. Okay. All right. I know that's not the easiest thing in the world, and I know there's a lot going on with that. So I'm going to pause for a moment. I'm going to go back over it one more time, and then we're going to move on, okay? All right, so first, we have all of them in square orientation, and that square orientation is has less affinity for oxygen as compared to the circular orientation, okay? The first thing that happens is a single oxygen binds to a single alpha subunit. That, per the Knf model, induces a conformational change in the next alpha subunit. And specifically what it does is it pushes the iron back down into the plane of the rest of the hemoglobin molecule, which helps the second alpha subunit bind to another oxygen, okay? After that's happened, when you have two oxygens, both bound to both alpha subunits, that induces a conformational change in terms of a rotation of about 15 degrees. This rotation of about 15 degrees opens the active sites of both beta subunits. So we go from our tense state to our relaxed state per the MWC model. And from there, the third and fourth oxygens can bind to the beta subunits. And that's kind of the spiel for hemoglobin in the MWC and Knf models. It's not easy, it's fairly confusing, but it works. Okay. Right. So now we're going to shift gears and we're going to talk about fetal hemoglobin. And the reason that I call it shifting gears is because we're basically dealing with something almost completely different, because I'm going to go ahead and spoil it. It does not have beta subunits. Fetal hemoglobin is alpha 2, and let's see, I never know how to draw gamma correctly, but we're just going to kind of like that. Oh, gamma 2 subunits. Okay, so it replaces the beta subunits for gamma subunits. But why does that change anything? And the reason that changes something, or to get to that, we have to answer what is 2,3-bisphosphoglycerate. Okay, so 2,3-bisphosphoglycerate is, by the most professional definition I can give you, a negative allosteric effector for hemoglobin. In other words, it's an inhibitor, right? So it destabilizes the deoxygenated form of hemoglobin, and it just kind of ruins it, right? So it makes it much harder to bind, and it reduces the affinity for oxygen. Turns out BPG, bisphosphoglycerate is abbreviated BPG. I didn't say that. So it turns out BPG tends to bind between beta subunits. Therefore, has a lower affinity for 2,3-BPG because it doesn't have those beta subunits, and therefore it has a higher affinity for oxygen. Okay, all right. So I'm shifting gears a lot more, and we're going to go kind of back to talking about inhibitors or inhibition, and talk about K systems and V systems. And we're pretty close to the end of this chapter, just so you guys are aware. So the reason I like to do all of that in one go is because we talk about MWC and KNF, but I don't really understand them until we've seen it, right? And so I think that hemoglobin example should go in there. Now let's talk about this, which is back to pretty much, normal inhibition topics. Well, not normal, but normal regulation topics. And then we're going to come back to, and then we're going to come back to hemoglobin and myoglobin. Okay, so this is, actually, let me start off by saying this first one is a K system, the second one is a V system. Okay, and the reason is a K system changes K and a V system changes V. Yeah, that's pretty self-explanatory. And specifically, I want to say, with the addition of some form of regulator, in this case, we'll use blue, either an activator or an inhibitor, and in most cases, either an activator or an inhibitor, K changes. So just a quick recap, K 0.5 or Km, whatever we want to call it, is the point at which 50%, we have basically 50% of the reaction rate, or if we were dealing with hemoglobin, it would be 50% binding, or whatever the case may be, okay? So Km is way over here when you have an inhibitor. It's way back here when you have an activator, and it's, in between the two when you have just the regular enzyme. Okay, so let's review what K0.5 or Km is. Okay, it's an indicator of affinity. But it's an inverse indicator of affinity. Okay, and the reason I point out the inverse part is low Km or low K0.5, high affinity, and vice versa. Okay, so when you have this really, really low Km, like you have with an activator, that's telling you it's binding a lot more quickly with less concentration. It's got a really high affinity. Whereas when you're dealing with the inhibitor over here, well, we're looking at it, it binds a lot more slowly, it takes a much higher concentration. That makes it an inhibitor. Okay, so eventually, as you guys can tell from the activator and regular, they level, like they reach the same Vmax, and eventually this would reach the same. They just cut it off before then. It would be slower than that, like that. But ultimately, Vmax unchanged. Okay, Vmax stays the same. K is the thing that's affected. K0.5 or Km, whichever you want to use. They say K0.5 because we're no longer dealing with Michaelis-Menten enzymes, but they mean the same thing. Okay, so that's a K system. In a V system, I'm sure you guys can guess, given that when we talked about a K system, K changed. A V system, V changes. It's complicated, right? All right, so let's think this through. V is obviously the maximum velocity, and we can see here that with an activator, it reaches a higher maximum velocity than neutral. With an inhibitor, it just goes really bad. However, it's really important to note here. So let's see. If this is approximately Vmax, then I don't know, maybe like here would be 1 over V, give or take. So that would make Km right about here. And I'm going to say Km just because I'm more used to it, but K0.5 is just as valid. Okay, switching colors to do the normal one, or the unaffected one. So Vmax is here. Half of that would be maybe here, and Km ends up being somewhere around there. So obviously I am not the best at approximating this. My lines are not always straight, but you guys can see that, you know, relatively somewhere to here, Km is relatively stable, right? It's not changing by drastic amounts. It ends up being about the same for all of them if I knew how to draw. And that's really what it comes down to. Km should be about here for all of them. And so obviously because I'm approximating, it's not, but Km is unchanged. Km, K0.5, whatever we want to call it, unchanged, right? I see questions about K systems and V systems not very regularly, but they're still covered, so I just very briefly wanted to go over them. The biggest parts of this chapter are recognizing those words, right? Regulatory enzyme, allosteric inhibition, feedback inhibition, allosteric regulator, and then obviously myoglobin and hemoglobin, and especially hemoglobin. There are so many questions about it, okay? This is a great question, okay? It is not allosteric. Why? Okay, well, I can tell you guys right off the bat, it does bind oxygen, it does contain iron, so we're really down to 2. So let's think about this in terms of hemoglobin. We know hemoglobin has 2 alpha and 2 beta subunits. Doesn't that make it a tetramer? And oligo means some subunits, few, so wouldn't a tetramer be an oligomer? And the answer is yes. So what the conclusion that we can reach is myoglobin is monomeric. Therefore, since each monomer has only one active site, in the case of hemoglobin and myoglobin, you only have one active site. Therefore, you cannot have that allosteric regulation, that cooperativity. There is no cooperativity in myoglobin because there's only one monomer. There's one monomer, which is very, you know, self-explanatory, it's repetitive. So there's a single monomer and with one active site, okay? All right, so that's effectively the end of the, or not the end of the chapter, or the end of the enzyme regulation chapter. We're gonna do one more, we're gonna work ahead just a tiny bit into the metabolism chapter. And the reason I like to do this is because metabolism is where everything comes together, and it's kind of the introduction that sets us up for the glycolysis chapters and stuff like that. And the glycolysis chapters, like I've said, are biochem. And so, we're just going to go ahead and jump in. So match the energy source with the metabolic classification of an organism that uses it for energy. In other words, a phototroph, a chemoorganotroph, a chemolithotroph, and an autotroph. That last, I don't know if it's a suffix, but that last part of each word, troph, is an organism using blank for energy. Okay, so photo is fairly self-explanatory. It means light. Okay, so a thing that uses light for energy is a phototroph. All right, plants, for example, phototrophs. Okay, we see chemotrophs or chemoorganotrophs and chemolithotrophs. So I think you guys can piece together based on organo that it's probably organic compounds, and you're exactly right. So the big thing here is chemotrophs of any variety use chemicals, right? They use compounds that are present, and then an organotroph uses organic compounds, and a lithotroph, or a chemolithotroph, I should say, uses inorganic. Okay, and that word litho, in my mind, it reminds me of lithium, right? And we've learned about we've learned about what's it called? Organolithium, things like that. Lithium is just kind of inorganic as a blanket term. Then finally, even if we didn't know the rest of it, we can piece together that CO2 is the autotroph. I believe the reason that the name is what it is because an autotroph, auto is kind of self. CO2 is very commonly a waste product, so it's basically saying it takes waste. And yeah, okay, all right. So metabolism is broken up into two processes, like it says here, catabolism and anabolism. Okay, and yes, there's one that's oxidative, one that's reductive, but I want to say the reason that they're oxidative, reductive, whatever, one, basically we eat and we break down what we ate. So I ate a sandwich, and that sandwich had bread, it had cheese, it had meat, whatever. I'm now going to break down the bread into subunits that I can use. So something like, stuff like carbon dioxide, stuff like water, stuff like glucose, the small pieces that we can actually use as building blocks. And then the other process is the one where we synthesize. So I have a bunch of glucose. I'm going to use that to make, for lipid storage. I have a bunch of, I just ate protein. I'm going to break those proteins down and use the materials to make whatever I need to make, and so on and so forth. Okay. And the way that I like to think of this, and the answer to this is that catabolism is oxidative, anabolism is reductive. Okay, oxidative, anabolism is reductive. So that's A. So I think that it's sometimes confusing to think. Okay, so sorry, I should mention catabolism is breaking down, anabolism is building up. Okay, so when we think of breaking down, that's oxidative because it's giving us oxidized products, right? So catabolism oxidizes stuff, and then anabolism reduces it to give us energy. So the reason anabolism is reductive is because it's reducing power, reducing potential, as I think of it, potential as energy. Okay. And the reason that catabolism has to be oxidative is to create that reducing potential. Okay? All right. So the goal of catabolism, we mentioned, is, going a little too far. The goal of catabolism is to break down products. The technical, very, sciencey, wordy definition is it's the oxidative I was going to say degeneration, that's not correct. Let me restart. It's the oxidative degradation of complex organic nutrients to simpler, oxidized, energy-poor products. Okay? So, and we're going to go into some details later, but the reason they have to be energy-poor, let's think about that. If they're energy-poor, that means we got all the energy out of them. Okay? And that energy is in our reducing agents, in the things like NADH and NADPH, which can be reduced and give us that power. Okay, so basically it's to make ATP, NADH, and NADPH. These are three of the most common things that we can reduce to get energy, for energy, and break down food, right? We eat something, break it down, make those, okay? Anabolism, and again, I'm gonna give you guys the wordy, complex definition, and then I'm gonna give you my definition. So anabolism is a reductive synthetic process in which complex biomolecules are synthesized from simple precursors and using ATP and NADH, okay? So I think of it as using small building blocks in parentheses plus energy from ATP, NADH, etc. And really this one should be NADPH to make our body, really like to make our body. So in other words, catabolism is opening up the Lego set and taking all the pieces out, finally separating every piece from everything else so that we can build. Anabolism is putting those pieces together in the way that we want them to be put together, okay? All right. So here's an example, or not really an example, here's the diagram of that. So we start with reduced fuel. So this is, you know, the foods we eat, and we oxidize them into the oxidized product. So this is, you know, basic stuff like water, carbon dioxide. Carbon dioxide is really common. It's a big oxidized product. And in doing so, we reduce NADPH, there we go, to NADP, or I'm sorry, NADP plus to NADPH. This is how we build, right? And you're going to see mostly NADPH in Biochem 2. We talked about it very briefly in Biochem 1. But yeah, we can use this NADPH in those reductive biosynthetic reactions that you see down there in anabolism to create our biomolecules that we need, okay? All right, and then as you guys can tell, there's one more question here, and we have about 8 minutes, so we'll be done in the next, I think, 3 minutes, okay? So why are our metabolic pathways regulated? Okay, many steps are near spontaneous in both directions and can react quickly. I don't know what that means. The synthesis of ATP needs to be regulated so as to not allow unwanted reactions. Okay. Many catabolic and anabolic pathways use the same enzymes. Okay. Our metabolic pathways are not regulated. So right off the bat, I'm going to say D is wrong. Okay. So I'm going to use an example from a little bit later into Biochem 1. In, I think, 3 weeks, 2 weeks maybe, you're going to be seeing this. And that example is glycolysis. So glycolysis is 10 steps. Okay, and we're not going to go over every step. We will before the third exam, but I don't want to waste your time right now focusing on something you're going to learn in class. Glycolysis is 10 steps. 10 steps with 3 very spontaneous reactions. Very spontaneous reactions. Okay, gluconeogenesis, which I'm going to say is kind of the opposite of glycolysis. It's not quite, but it's effectively making glucose from pyruvate, which is the end result of glycolysis, is 11 steps, and it has 7 of the same enzymes as glycolysis. Glycolysis doing the reverse reaction. Reverse reaction. In other words, if glycolysis is the green bit, and it's actually, so we have A to B to C to D. Glycolysis is the green. It's going forward, and it's using E1, E2, and E3. Well, gluconeogenesis can do the same thing using the same enzymes for most reactions. There are obviously some exceptions, and we're going to talk about those when we get to these two pathways, so that I'm not, forcing you all to learn ahead. But effectively, what I'm saying here is, for the reactions that are near spontaneous in both directions, I can make B, and then B could go back to A, or it could form C, right? So we need to shut down one of the pathways. We need to say, hang on, let's turn off the red pathway. Stop the red one, and we're going to say, go green. Okay, I was trying to draw a check mark. It wasn't the best. Okay, when we do that, we need to regulate the red pathway and let the green go. So we need to inhibit red and activate green so that we get product D. If, on the other hand, we did the opposite, so we let red go and we actually also kept green going, D would go back to form C and then go back to B, and B would go back to form C and then D, and it would go forward and backwards all at once. And we would get a bunch of just forward and backward reactions. And so it would be chaos, right? We would never get what we actually need. And so the answer to this is actually the steps in each are, not super spontaneous, right? And when I say that, what I mean is, for example, if it was -2 kilojoules per mole, if that was the delta G value, the free energy, yes, it's spontaneous, but when we think of ATP hydrolysis, -30, -31 kilojoules per mole, I mean, we're talking about very spontaneous versus, kind of, it'll go in that direction, but very slowly. You can pair that with the next reaction down the line, which may be spontaneous in the opposite direction, and you can make it go against that -2 number. -30, -31 number, it's not going to go backwards, right? And again, I don't want to jump into too much detail because I don't want to waste your time with glycolysis yet. I believe we're going to be talking about that a tiny bit next week, mostly in week 10. But yeah, so that's kind of the reason is, if we have many steps in that can go in both directions, we want to shut down one direction. Okay, all right. And that's actually all that we have today, and actually basically right on time. So if you have any other questions, feel free to come in to drop in tutoring. We're open at 11 a.m. to 8 p.m. Monday through Thursday, and 11 a.m. to 5 p.m. on Fridays and Saturdays. And we'll be able to help you out anytime. Okay. Hope you all enjoyed. I hope you all learned something, and feel free to ask any questions you have. Thank you.